Welcome to the Deep Dive, where we take a look at the latest happenings in genomics and bioinformatics. We've got a really interesting collection of articles and blog posts today. Everything from brand new technologies to important research breakthroughs.
Yeah, it seems like every day there's something new and exciting happening in these fields.
For sure. Let's jump right in and start with this new product that really caught my eye. Olink Reveal. It's supposed to make targeted proteomics much more accessible.
Oh, yeah, I read about that. So targeted proteomics is all about measuring specific proteins, the ones you're interested in. And usually that requires a lot of, specialized equipment. You'd normally only find that kind of setup in a big lab or a genome center.
Right.
But this Olink Reveal, it seems to simplify things a lot. It focuses on a panel of about 1,000 proteins.
Right.
And they've chosen proteins that we know are connected to human variation and disease. So researchers can get a good look at a targeted set of proteins without needing a ton of resources.
It's almost like a curated toolkit
Exactly.
for exploring this specific part of the proteome.
Mm-hmm.
Researchers could use it to investigate specific pathways
Yeah.
or disease mechanisms,
Yeah.
maybe even potential drug targets.
Definitely.
That's really cool. I mean, if this really takes off, we could see a lot more research in things like personalized medicine and drug development, too.
Yeah, I think so. Imagine all these smaller labs and research institutions suddenly being able to do this cutting-edge proteomics research. It could lead to a lot more discoveries and maybe even new therapies.
Yeah, that's a really exciting prospect. But it does make you wonder, if they're just focusing on this specific panel of proteins, what about all the other proteins? Could some of those be important, too?
That's a great point. I mean, targeted approaches like this Olink Reveal, they're definitely efficient. You get a really focused look at what you're interested in, but you could be missing some crucial information that's outside of that targeted set.
Right.
So those larger scale untargeted proteomics approaches, those are still going to be really important.
So it's all about choosing the right tool for the job. Okay, so we've talked about the human proteome, but what about plant genetics? There was this really interesting study about how soybeans defend themselves against nematodes.
Oh yeah, those tiny little worms.
Yeah,
They can cause a lot of damage to crops.
for sure. But it turns out that soybeans have some pretty sophisticated defense mechanisms.
Right. The researchers used RNA sequencing to analyze how soybeans respond to those nematode infestations. And they found that the soybeans actually activate specific genes.
Oh, okay.
And these genes produce enzymes that can actually break down the nematode's cell walls
Wow.
or disrupt their development.
So it's like an evolutionary arms race. I
Exactly.
mean, soybeans are a vital crop all over the world, so understanding how they protect themselves could have a huge impact on food security.
Absolutely.
Right.
This kind of knowledge could be really useful for breeding programs
Right.
and for coming up with better pest management strategies, ultimately leading to crops that are more resilient and productive.
Now let's move from plants to the human gut. There's a study about the immune cells in the gut microbiome.
Ah, the gut microbiome, that incredible complex world inside us.
Yeah, and it turns out there's a lot going on in there. This study used RNA sequencing to take a close look at how immune cells interact within the gut.
Right.
They focused on a specific type of T cell that plays a crucial role in defending the small intestine against pathogens.
Right. And what they found is that these T cells, they don't just sit in one place, they actually move around throughout the gut.
Wow.
They interact with other immune cells and they adapt their defenses depending on the environment they're in.
So it's almost like a microscopic security force, constantly patrolling and responding to threats.
That's a great way to put it.
This kind of detail, it wouldn't have been possible to observe before. But thanks to RNA sequencing, we're really getting a much clearer picture of how the immune system works at the cellular level.
Absolutely. And this research has implications beyond just gut health. I mean, the gut microbiome influences so many things in the body, digestion, even mental health. So understanding how it interacts with the immune system is crucial if we want to develop new therapies and interventions.
Now, speaking of understanding complex interactions, there's this whole field of multi-omics that's gaining a lot of traction. It's all about integrating data from different biological levels, genomics, transcriptomics, proteomics, and more to get a more holistic view of how biological systems work.
It's like putting together a giant puzzle.
Yeah.
But instead of a static image, we're building a dynamic model of a living system.
Right.
By combining data from different sources, we can see connections and patterns that you wouldn't be able to see if you were just looking at one type of data.
For example, researchers are using multi-omics to try to find new biomarkers for diseases, or to predict how an individual might respond to a specific treatment, and of course to develop personalized medicine strategies.
Right.
This integrative approach is so powerful. I think they have huge potential for improving healthcare and really understanding the complexities of human health.
I agree. And as we move forward, the tools and techniques we use to analyze this data, that's going to be crucial. That's where bioinformatics comes in.
Absolutely. Bioinformatics is essential for processing, analyzing, and interpreting all the data we get from these new technologies and research approaches, from targeted proteomics to RNA sequencing and beyond.
Yeah, speaking of new bioinformatics tools, there's one called DICAC. It's a bit of a mouthful. It stands for droplet-based single-cell high-C.
Okay, that is a mouthful.
Oh.
So what does it do? Why is it important?
Well, it's all about understanding the 3D structure of DNA inside a cell's nucleus.
Okay.
See, it's not just the sequence of DNA that matters, it's also how it's folded and packaged. This 3D structure affects which genes are turned on or off, and it can be very different between cell types. So this DSIC, it lets us map this 3D structure at the single cell level. It gives us a much better understanding of how gene regulation works in different tissues and cell types.
So it's like having a 3D map of the genome.
Right?
That's incredible.
And this knowledge is going to be incredibly valuable for developmental biology, cancer research, even regenerative medicine.
Wow. And RNA sequencing is being used to study the genetic diversity of phages, right?
That's right.
Those viruses that infect bacteria.
Phage research is really fascinating, especially now with all the interest in the human microbiome.
Right.
Because these viruses, they play a huge role in shaping bacterial populations inside us.
Okay.
Which in turn affects our health in a lot of different ways.
So it's like a hidden universe inside us.
It is. And understanding these interactions is so important if we want to figure out how to manipulate the microbiome for therapeutic reasons, like using phages to target specific bacteria that cause disease.
Okay.
Or to encourage the growth of beneficial bacteria.
So the phages are like our little allies.
You could say that.
Now, one more thing that's really interesting is the role of artificial intelligence in bioinformatics. There are companies like DeepSeek and Olink that are doing some really cool things.
Right.
They're using large language models, or LLMs, which have been incredibly successful in natural language processing. Now they're being applied to the challenges of bioinformatics.
It's amazing what they can do. They can be trained on huge amounts of biological data, like protein sequences, DNA structures, scientific literature.
Yeah.
And they can learn patterns and make predictions.
So it's like having a digital research assistant that can analyze these enormous data sets.
Exactly.
It can help researchers identify new drug targets, predict disease risk, maybe even design new proteins.
And with companies like DeepSeek and Olink leading the way, we can expect to see even more incredible applications of LLMs in genomics and bioinformatics.
For sure. LLMs have the potential to revolutionize how we approach drug discovery, personalized medicine, even our understanding of the very basics of life. All right, let's shift gears a bit and talk about some industry news that could really shake things up.
23andMe, the company that does those direct-to-consumer genetic tests.
Right.
They just announced that they're going to be offering something new for research.
Oh, really?
So it seems like they're moving beyond just giving people information about their ancestry and health. They actually want to contribute to scientific discovery.
That's interesting. I mean, they've gathered a massive amount of genetic information from their customers. So what could that mean for research if they start using that data?
Well, I think it could be huge for large-scale genetic studies and for personalized medicine, too.
Right.
With all that data, researchers could gain some really important insights into the genetic basis of diseases. They could find new drug targets and even develop better ways to diagnose and treat people.
So they're going from a consumer-focused company to a major player in research. It's a big shift.
Yeah, definitely. And speaking of major players, GSK, the pharmaceutical giant, just made a huge acquisition in the biotech space.
Oh, wow. What did they acquire?
They bought a biotech company for $1 billion.
$1 billion. Wow. That's a serious commitment.
Yeah, it shows you how serious they are about innovation in the biopharmaceutical sector.
So what do you think this acquisition tells us about GSK's strategy and maybe even the future of the whole industry?
Well, it seems like they're really trying to boost their research and development capabilities, especially in areas like gene therapy, precision medicine, and new drug discovery platforms.
Okay.
It's a clear sign that biotechnology is becoming more and more important for the pharmaceutical industry.
So GSK is going the acquisition route, but other companies are focusing on partnerships and collaborations. Absci, a company that specializes in AI-powered drug discovery, just announced a partnership with Okin. They're experts in federated learning and data privacy.
Oh, that's an interesting pairing.
Yeah, I thought so too. It really highlights how important data sharing and collaboration are becoming, especially now with all this big data.
Right. Combining Absci's AI with Okin's federated learning could lead to some major advancements in drug discovery.
Absolutely. And it seems like they're really thinking about the ethical and regulatory issues too.
Right. Data privacy is a big concern.
So how does federated learning work exactly? I mean, how does it benefit drug discovery research?
Well, with federated learning, AI models can actually be trained on data that's spread out across multiple institutions.
Okay,
But they don't have to directly share the raw data.
I see.
So researchers can access and analyze these huge, diverse data sets, which is really important for developing effective and personalized therapies.
Right.
But they can do it without compromising data security or patient privacy because the sensitive information stays where it is.
It's like a global network of research collaborators.
Exactly.
That's really cool. Now, in other news, Charles River Laboratories launched a new cloud-based platform for managing lab animals.
I saw that. It's a cloud-based vivarium platform. It's a big step towards modernizing how we manage lab animals, and it could really improve animal welfare and research efficiency, too.
I think so too. I mean, by using cloud technology, researchers can keep track of animal health, manage breeding programs, and analyze data much more effectively.
Right.
So potentially we could see more reliable and humane research practices.
Hopefully.
And this move towards digitalization in animal research, I think it could have a big impact on the entire drug development process.
Yeah, it could.
It could potentially speed up the discovery and development of new therapies. Now let's talk about RNA sequencing analysis. The RNA-seq blog has highlighted some upcoming workshops and webinars. They're all about helping researchers get more insights from their RNA sequencing data.
That's great.
It seems like these events could be really valuable, especially for researchers who are new to RNA sequencing.
Yeah, or for those who just want to stay up to date on the latest analysis techniques.
Right, exactly. So what kind of topics are they covering?
Well, there's a webinar called Transforming Pipeline Outputs into Biological Insights. It's all about helping researchers bridge the gap between running those bioinformatics pipelines and actually extracting meaningful biological information from all that data.
Yeah, that's a crucial step.
And then there's a workshop called Intermediate RNA-seq Analysis using R. It's hands-on training for analyzing RNA sequencing data using the R programming language.
Oh, that's great. R is such an important tool for anyone working with genomics data.
It is.
That workshop could really help researchers learn how to do more sophisticated analyses and create better visualizations,
Yeah,
which would help them understand their research findings even better.
definitely. And there's also a blog post that gives a really good overview of single-cell RNA sequencing analysis.
Oh yeah, single-cell RNA sequencing has been a game changer. It lets us study gene expression at the individual cell level.
Right. It reveals all that hidden diversity within tissues and organs.
No, we never knew about before.
Exactly. So this blog post is a great resource for anyone who wants to learn more about this technology and how it's being used.
It sounds like there's something for everyone, no matter what their level of expertise is.
Yeah, I think so.
Now, getting back to multi-omics, there's this new tool called MOSCOT.
I read about that. It stands for Multi-Omics Single-Cell Optimal Transport.
Yeah.
It's a computational framework that lets researchers integrate different types of single-cell data, like gene expression data and protein abundance data.
Okay.
So you can get a more complete picture of cellular states and how cells change over time.
Interesting.
It uses a mathematical approach called optimal transport to align and compare data from different sources.
Okay.
So you can see the connections and patterns across different biological levels.
It sounds really powerful. How are researchers actually using this kind of integration in their work?
They're using MOSCOT and other multi-omics approaches to study a lot of different biological processes, cancer development, immune responses, the aging process.
Right.
By combining all this data, we can really start to understand those complex interactions that drive these processes. And hopefully we can find new targets for therapeutic interventions.
It seems like multi-omics is becoming more and more important for figuring out how these complex biological systems work.
Absolutely.
And of course, we can't forget about artificial intelligence. It's making a big impact in bioinformatics. We've covered a lot of ground today: new technologies, research breakthroughs, industry shakeups, and of course, the growing influence of AI.
It really is an incredible time to be following genomics and bioinformatics. Everything is moving so fast. and we're making real progress in understanding life at the molecular level.
So as we wrap up this deep dive, I'm curious to hear your thoughts on the bigger picture. What are the key trends and challenges that you see shaping the future of this field?
Well, one of the most important trends is that these powerful tools and technologies are becoming much more accessible. Like that Olink Reveal we talked about, it makes something like targeted proteomics available to a much wider range of researchers, and that could really accelerate discoveries and lead to more innovation.
And that accessibility is so important for creating a more inclusive and collaborative research environment. What about the role of data? We're generating more data than ever before in genomics and bioinformatics. Seems like it's almost overwhelming.
Absolutely. We're drowning in data. And that's both a good thing and a bad thing.
Right.
It's a huge opportunity to uncover new insights, but it also presents a big challenge when it comes to storing, processing, and analyzing all that information.
And that's where bioinformatics comes in. We need robust computational tools and methods to make sense of all this data.
Exactly. And thankfully, there have been some really exciting advancements in machine learning and artificial intelligence that are helping us tackle these challenges.
Yeah, like those large language models.
Right.
They can be trained on massive biological data sets to do things like predict protein structures or identify drug targets. They can even design new proteins with specific functions. It's almost like having an army of digital research assistants at your fingertips.
It is pretty amazing.
So for our listeners who are really into this stuff and want to stay ahead of the curve, what advice would you give them?
Never stop learning. Stay curious. Engage with the scientific community and explore all the resources that are out there online, workshops, conferences. Genomics and bioinformatics are always changing, so you have to keep learning and adapting.
Great advice. And with that, we've reached the end of our deep dive into the latest genomics and bioinformatics news. We've covered a lot today, but there's always so much more to discover.
We've just scratched the surface. This field is full of exciting discoveries just waiting to be made.
So keep exploring, keep learning, and who knows, maybe you'll be the one to make the next big breakthrough. Thanks for joining us, and until next time, keep pushing the boundaries of science.